Okay, so genetics, you're going to get like 10 or 15 questions from this topic, so it's very important to remember. So first of all, you should know the basics. Then they can ask you about different techniques, diagnostic procedures, like we collectively call them NIPT. Then they will ask you about the most suitable investigation for any given condition. You should know about that. Then what are the how much time a certain investigation is going to take, you should know about that. Then how you will convey the results, and how you will break a bad news for the mother also. And you should remember the very specific things. Okay, so structure of chromosomes, why it is important, because DNA, that is the basics, that is the basic material which distinguishes different species. So DNA is, this is our genetic material which is contained in the chromosomes. Okay, so that's why important to understand the structure of chromosome. So how, so which technique do we use to find that someone is having an abnormal DNA? Karyotyping. You should remember there are certain screening tests. Screening tests means that we are just screening someone, we are just testing if they have any condition. And then we are doing definitive tests or diagnostic tests. Diagnostic tests are totally different. They will confirm or exclude a certain diagnosis. So the most popular technique to the date is karyotyping. So sometimes they will ask you which material can be used for karyotyping. So this technique utilizes peripheral blood lymphocytes and can still be performed on samples received in the lab after more than 24 hours after sampling. It can also be performed on chorionic villi, fetal cells from amniotic fluid, and fetal blood following fetal blood sampling. Results are available within 48 to 72 hours from fetal blood and from chorionic villi also in 48 to 72 hours, but if you will request amniotic fluid, then results will be available in 2 to 3 weeks. So important to remember these timelines. Cell division is arrested in metaphase by the addition of colchicines, which prevents spindle formation. Chromosomes are typically stained with Giemsa stain, and we call it G banding also. The bending pattern of each chromosome is characteristic with a dark band containing mainly inactive genes. Other bending patterns, they can be obtained using quinacrine, then we will call it Q banding, and visualized by ultraviolet light. C banding to show highly repetitive microsatellite repeat DNAs at the centromeres. Centromere, where the short and the long arm of the DNA, they are uniting. So we will talk about detailed structure of DNA in a while. Just remember that bending allows the identification of missing or additional chromosomes, because then things will be highlighted, so you will be able to see them clearly. If we look at the structure of DNA, it contains chromatin. So DNA exists in the condensed chromatin form to fit into the nucleus. DNA loops twice around a histone octamer, which is a protein octamer, to form a nucleosome, and that's why it will have the appearance of beads on a string. H1, which is histone 1, histone can be of 4 types. So H1 binds to the nucleosome and to the linker DNA. This is the linker DNA, which is linking them, thereby stabilizing the chromatin fiber. So phosphate groups, they give DNA a negative charge. Lysine and arginine, they give group, they give histones a positive charge. So DNA, so positive and negative charges, they will bind with each other tightly. In mitosis, DNA condenses to form chromosomes. DNA and histone synthesis, it occurs during S phase of the cell cycle or cell growth. Mitochondria, they have their own DNA which is circular and does not utilize histones. So histones are seen only in the regular DNA. Then if we talk about the heterochromatin, on electron microscope, it appears darker as it is labeled like H in the figure. So transcriptionally, this is the darker one labeled as H. Transcriptionally inactive, and this is sterically inaccessible, and it will increase methylation. It will increase with methylation and decrease with acetylation. Two processes which will make it more active or inactive. You can remember that heterochromatin, this is highly condensed, and bar bodies, these are inactive X chromosomes and they may be visible on the periphery of the nucleus. Euchromatin. Euchromatin, as denoted by this letter E, this is less condensed, appears lighter on electron microscopy, and this is transcriptionally active, and this is sterically accessible. So U, actually the word U means true. So it means truly transcribed. And you can remember that euchromatin is expressed. Then sometimes you can find this in the questions, DNA methylation. So DNA, if methyl is attached to DNA, it changes the expression of a DNA segment without changing the sequence involved with genomic imprinting. X chromosome inactivation, repression of the transposable elements, aging, and carcinogenesis. So DNA is methylated in imprinting, and methylation within gene promoter, this is typically, represses gene transcription. And remember that methylation makes DNA mute. These are, you should have clear concepts. Okay? Methylation will make it mute. Now, what if a histone is methylated? Histone methylation, it usually causes reversible, right? Transcriptional suppression, but can also cause activation depending upon location of methyl group. Histone methylation mostly makes DNA mute, mostly, but not always, mostly. Histone acetylation relaxes DNA, it relaxes DNA coiling, allowing for transcription. So histone acetylation makes DNA active. So A for active and M for mute. You can remember like this. Okay. So then you should remember that if you are testing this material, that banding allows the identification of missing or additional chromosome material of at least 4,000 kilobytes. If it is smaller than that, then maybe we cannot identify that. So we will talk about mutations in the DNA after a while, but first you should know about the chromosome heterochromatomorphism. So chromosome heteromorphism. There are variations in the amount of DNA present in particular chromosomes from different individuals caused by variation in the size of repetitive DNA, and they are inherited rather than acquired, which we will take from the environment, and these changes they occur after birth. Y chromosome, it shows the greatest variation, while the X chromosome shows the least variation. There are four main types of groups of heteromorphism. One, the size of YQ, the long arm of Y chromosome. Q is the long arm and P is the short arm. So 10% of normal males, they have a YQ which is obviously shorter or longer than usual. Then size of centromeric heterochromatin. Then satellite polymorphism, which means variation in the size of the satellite in acrocentric chromosomes. Which are acrocentric? Acrocentric where the this centromere is located towards one end. So yeah, the larger one like that. 13, 15, 21, 22. So metacentric chromosomes, they do not have satellites. Then there are fragile sites. Fragile sites, these are constriction sites other than the centromere which are prone to breakage. There are at least 80 common fragile sites. Most are not associated with any clinical syndromes except the fragile site at XQ27.3, which is associated with learning disability, which is Fragile X syndrome. And why that is important? Because that is the second most common uh, cause of this mental retardation. We will talk about replication and all of these concepts. As you know that this is like double helix. Then some enzymes like helicase, it will come and it is like the zipper on a zip, and it will divide it into two parts. So replication will take place like this. Okay, replication will be there, different enzymes will be there, then two strands are there, and then one will contain one base, so appropriate base will come and attach here. Like you can see that one is brown and one is very this light of like this fawn color. So they will match with their appropriate pairs, and then DNA polymerase free, these enzymes will come and they will repair them, and then this replication fork will go up. It will keep opening them, and then through this method of this DNA development synthesis, everything will come and they will attach the bases, and then it will be, the DNA is, as we know it is developed in small fragments like Okazaki fragments, and then they are united by some other enzymes. So we will discuss this one in more detail later. So this is just to give you a concept. And the appropriate bases will come and they will complete one strand of DNA, which will have a double structure. So if we talk about DNA structure, then nucleic acids, they can exist as deoxyribonucleic acid or ribonucleic acid. So these are made up of a chain of nucleotides. Each nucleotide contains a nitrogenous base, a sugar molecule, and a phosphate group. The base is either a purine or a pyrimidine. Purines mean adenine and guanine. Pyrimidines, they mean, yeah, and pyrimidine means cytosine, thymine, and uracil. So thymine occurs in DNA only, while uracil occurs only in RNA. Some questions can be so simple, like just remember thymine has been replaced by uracil in RNA. The sugar molecule is either ribose or deoxyribose, and they are held together by a 3 to 5 phosphodiester bond. The DNA molecule is made up of two chains of nucleotides held together by hydrogen bonds between the bases. A purine in one chain is always paired to a pyrimidine in the other chain. ATGC. Okay. Yeah, so that's how they will stay. Sometimes they will give you a template, a strand in the question, and they will ask you what should be the sequence of bases. So just remember, A is going to be with T and G is going to be with C at GC. So adenine, thymine, and guanine and cytosine. So the chain end terminated by the 5 carbon of the sugar molecule in the 5 end, and this always lies opposite the 3 end of the strand, and the two chains are arranged in a double helix. So DNA replication, it occurs during the synthesis phase of the cell cycle. The double helix is separated by the enzyme DNA helicase at different sites on the DNA molecule. DNA synthesis is undertaken by DNA polymerase in the 5 to 3 direction. The leading strand is synthesized as a continuous chain, while the lagging strand is synthesized in pieces called Okazaki fragments and then joined by the DNA ligase. As I showed, I think we talked about this, that one, can you see that one strand is continuous? Okay, so this is the leading one. Why the leading one? Because the sequence of the bases here is going to dictate which second base will come here. Okay, so this process, you can see here, here is the helicase. Okay, it is helicase is going to open this all of this stuff. Then you can say that this is the leading strand, this is and the leading strand and the lagging, this is the leading one, this is the continuous, okay, and the lagging one, the one which is following the sequence. Then small bases are formed, nucleotides are forming in small fragments, Okazaki fragments, and then the primer, it is bringing them here and they are attaching, and then they are under the effect of DNA polymerase, DNA ligase, it is going to repair any, if there is any defective thing, mismatched pairs, then it is going to remove them. So it is like a quality control procedure, okay? And it is going polymerase means it is going to join them. Then DNA polymerase one was here and 2 is here, okay? Primer, it is conditioning them, preparing them for their final stage, and here the replication fork is going on. So the leading strand, if it is in direction 5, then the lagging is will be in direction 3. Okay. Here the leading strand was in the 5 direction, and then the opposite came in 3 direction. Here it was in the direction started at number 5, so it is in 3 direction. So that's how you can always interpret that how they will behave. Okay. So then only one chain of double helix is all newly synthesized, and the DNA replication is therefore semi-conservative. Semi-conservative because we are keeping some element. That's why the generations they are and the species they are maintained with the same genetic composition. So transcription is the synthesis of messenger RNA from DNA. It's not from DNA, from messenger RNA to DNA. It is from DNA to messenger RNA. So as we talked about chromosome structure, primary coiling of DNA into double helix, secondary coiling of double helix around spherical histone beads to form nucleosome, tertiary coiling of nucleosomes to form chromatin fibers. Chromatin fibers, they form loops on a scaffold of non-histone acidic protein. Chromatin fibers, they are bound into a tight coil to form chromosomes. All chromosomes, they have a centromere, which is the site of attachment of the spindle apparatus during cell division. Here the dividing chromosome spindles, they will arrange themselves. So metacentric, it has these chromosomes, they have the centromere in the middle. Which chromosomes are metacentric? 1, 3, 16, 19, and 20. Ectocentric, they have the centromere close to one end. Ecto means the end, periphery. And these are the chromosomes usually involved in balanced translocation 13, 14, 15, 21, 22. So they are easy to remember, okay? And you will see them in balanced translocations, and the most common condition will be, yeah, Down's syndrome. So submetacentric, they have a centromere in between which, in between the end and the center, okay, in between the end and the middle. So that will be a submetacentric. And chromosomes, they have a short arm, P, petite, and a long arm or Q arm. Histones, we talk about them. We have seen this one before. Histone 1, basic nuclear protein around which the DNA molecule is tightly bound. It makes up about 50% of the mass of a chromosome, and this is rich in lysine or arginine, and 25%. And lysine and arginine, these are basic amino acids. The gene coding for histone proteins are clustered and randomly repeated 30 to 40 times in a human. This normal repetition is important, otherwise things can go abrupt, and they can cause different abnormalities. So that's the normal pattern, that how chromosomes should divide. Then the question arises that how mutations in the DNA can take place. So if we think about because the mutation in DNA means that some things will be changing, so DNA is going to be abnormal. So if the DNA is abnormal, the coding has been inappropriate or whatever was the cause, then product will not be as it is meant to be, right? So that's why abnormalities will develop. If we talk about the severity of damage, then we should know silent mutations, less harmful than the missense. These are less harmful than the nonsense, and they are less than the frame shift. 4 points: silent, missense, and nonsense mutations. The transition is, it can be from purine to purine, for example, A to G or G to A, or pyrimidine to pyrimidine, for example, C to T. Then transversion, like purine to pyrimidine, because now the type of base is also changing, for example, from A to G, or pyrimidine to purine, like C to G. Then what are silent mutations? Here, nucleotide substitution will take place, but it codes for the same amino acid. Often base change in third position of codon. So like transfer RNA, it will wobble. So the base will be changed because amino acids are like that several amino acids, they will have the same function. So that's why they said silent, because only if you are doing this karyotyping due to some other condition, then you will realize that there is some abnormality, because as such on their own they are not going to cause any disease. Then missense, nucleotide substitution resulting in changed amino acid, and this is called conservative if new amino acid is similar in chemical structure. One example is in sickle cell disease, substitution of glutamic acid with valine. So ideally glutamic acid should have been there, but now valine has replaced it. Then another form of mutation can be nonsense. Here, nucleotide substitution resulting in early stop codon. What are the stop codons? You should remember UAG, UAA, UGA. These are the stop codons, and they will usually result in non-functional protein. So what will happen? They will cause nonsense mutations. Then frame shift, deletion or insertion of a number of nucleotides not divisible by 3, resulting in misreading of all nucleotides downstream. Protein be shorter or longer, and its function may be disrupted or altered. So, for example, Duchenne muscular dystrophy or in Tay-Sachs disease. So the whole of the this frame has been shifted. Okay, frame of the nucleotides has been shifted. So there is an abnormality. So from the level of this abnormality, level of this change, below that one, everything will be disrupted. Then supply site. Mutations are at a supply site, and as a result retained intron in the messenger RNA. Intron means inactive RNA, DNA. Retained in the messenger RNA and protein with impaired or altered function. And rare cause of cancers, dementia, epilepsy, and some types of beta thalassemia, these supply site mutations will take place. So you should just have a basic idea that how mutations can take place. So, and then you should remember that what are the steps and from the synthesis of DNA, and we will talk about them in detail once we will do microbiology. It is very important. Autosomal modes of inheritance. As you know, there can be different modes, so very important to understand the basic concepts. And this one is very helpful because you can get very easy marks. Like what? Like if you will see, they will ask you simple questions like what is the mode of inheritance. So these are very easy questions. Okay. I will pause just for 1 minute and I'll be back. Just 1 minute, please. Okay. If we talk about modes of inheritance, there are different modes like autosomal dominant inheritance, autosomal recessive inheritance, X-linked dominant, and X-linked recessive. So here I have brought all of these together for the sake of comparison, but we will keep referring to this one as we will go through the lecture. Autosomal dominant, as you can see, this black color, it means the affected individuals. Okay, so autosomal dominant conditions, they are often due to defect in structural genes. Many generations will be affected. Both males and females are affected. You know, these circles, they denote females, and squares, they mean male. So you can see it is going in a vertical way, and both genders are affected. Often they are pleiotropic, which means multiple apparently unrelated effects, and variably expressive. Different between, they will have different traits between different individuals. Family history is crucial to diagnosis. With one affected parent, on average half the children affected. As we will see in detail, we need just one copy of the gene to inherit a disorder. So that's why there is no skip generation. Okay? So each generation is affected, both genders are affected, and we need just one copy of the defective gene. And one parent is affected, half of children will be affected. Second mode of inheritance is autosomal recessive. Here you can see they both are fine. They produce more children. So they both were carriers. Okay. So that's why one baby is affected and three are unaffected. So then in the next generation, nobody has expressed this disease. So if and this type of disorder needs two copies of the gene. So one should come from mother and one should come from father. Still one child will be affected and others, they can be 50% can be carried. And then there is a skip generation. No one expressed disease here and no one has expressed here, but we have seen it in this generation. So autosomal recessive, often due to enzyme deficiencies and usually seen in only one generation. Commonly more severe than dominant disorders, patients they often present in childhood. Increased risk in consanguineous families where they are marrying them in the same family. With two carriers or heterozygous parents, on average, 1 out of 4 children will be affected. So homozygous with homozygous, and half of children will be carriers, and 1/4 of the children they will be neither affected nor carriers. So one will get a green signal that nothing is wrong here, but others two will be carriers and one will be affected. Another mode of inheritance can be X-linked recessive. So as you can see, let's assume these were carriers. Okay, this one is affected, and then again, next generation again, this male has been affected. Male was affected here, male is affected here. Females are carriers, but they have not expressed the gene normality. Sons of heterozygous mothers, they have a 50% chance of being affected. No male to male transmission, skip generations. Why no male to male? Because this is X linked. Males, they don't have, they will transmit only Y genes. So they will not pass the disorder. But the affected female will not be expressing the disease because she is only a carrier and she is passing it on to the next generation. So commonly more severe in males, and females usually they must be homozygous to be affected, so which is very rare. So if they give you a question that the mother is a carrier, uncle, her brother was also died due to hemophilia, so what is the chance that her sons will be affected? So half of the sons will be carriers because she has two eggs, so both are not carrying the defect, one will be carrying the defect. Okay, so all daughter, half of the daughters they will be carriers and half of the sons will be affected. While if a male has hemophilia, then all of his daughters will be carriers and none of the sons will be affected because this is X-linked recessive transmission. So the father will pass one gene, so the girls will be carriers but they will not be affected. And this type of questions they will ask you in part 2 also. Okay, so the next mode of inheritance is X-linked dominant. So this is transmitted through both parents. Mothers transmit to 50% of daughters and sons, and fathers, they will transmit to all daughters but no sons. This is X-linked dominant. So X-linked dominant here, the father was affected. So he passed the gene to one of the daughters and she passed it on to her son. Similarly, here one girl was affected, so it was transmitted to the, she had two girls but transmitted to just one girl, so 50% of the daughters and sons, they will be affected. Two sons, but again just one girl affected. Example will be, these are very, this type of disorders are relatively rare. One example will be hypophosphatemic rickets. Formerly it was known as vitamin D resistant rickets. Inherited disorder resulting in more phosphate wasting at proximal renal tubule, and it results in rickets-like presentation. Other examples are Fragile X syndrome and Alport syndrome. It is their favorite. Fragile X syndrome. What type of, what is the mode of inheritance? This is X-linked dominant. Okay. Just remember, with the, in the end, I think we have given you a list of the conditions and the mode of inheritance. Mitochondrial inheritance, this is also very important. You should remember that this is transmitted only through the mother, and all offsprings of affected females, they may show signs of disease. So variable expression in a population or even within a family due to heteroplasmy. So there can be mitochondrial myopathies. Here you cannot predict. This is coming from the maternal DNA, mitochondrial DNA, circular in form. So half of the children, they are affected. Okay? And then in the next couple, she has passed it on to both babies, and this one didn't pass on to anyone. So there is heteroplasmy. Like mitochondrial myopathies, these are rare disorders, often present with myopathy and lactic acidosis and CNS disease like Neil's syndrome. This is mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes. Okay, just remember meals. Secondary to failure in oxidative phosphorylation. Oxidative phosphorylation takes place inside mitochondrial DNA. Muscle biopsy often shows red fibers due to accumulation of diseased mitochondria. Second condition is the Leber hereditary optic neuropathy. Very rare. If they will ask you this question, they will ask only about the mode of inheritance. So mitochondrial inheritance in males and in Leber hereditary optic neuropathy. Cell death in optic nerve neurons and subacute bilateral vision loss in teenagers or young adults in 90% of the time. And going back to this autosomal dominant condition, this trait or condition is expressed in heterozygous individuals. Male and females are affected in equal numbers. Then vertical pedigree pattern, and doesn't skip generations. Males and females, they can transmit the condition to their male-female offspring. Unaffected individuals cannot transmit the disease. The exception is in conditions with variable or non-penetrance, where individuals with the mutant allele, they have a normal phenotype but they can transmit the conditions. They are normal but they can transmit it. Through which, so which will be the phenomenon? It will be variable or non-penetrance. So variable means that one person, both several persons have inherited the disease, but they are showing different intensities of the disease. And non-penetrance, it means that they how how deeply or how severely an individual is affected. So they show autosomal dominant disorders, they show variable expressivity, both in terms of severity of the condition and age of onset. Affected individuals, they have a 1 in 2 risk of their offspring being affected. There is a paternal age of effect with respect to the incidence of new mutations. Common examples are autosclerosis, von Willebrand disease, familial hypercholesterolemia, Huntington's disease, tuberous sclerosis, myotonic dystrophy, neurofibromatosis, hereditary spherocytosis, and familial polyposis scoli. Regarding this von Willebrand disease, type 1, 2 autosomal dominant, 3 is recessive, but mostly it will be categorized into autosomal dominant. Then the next mode of inheritance, autosomal recessive, as we saw above, the trait condition is only expressed in homozygous individuals. Males and females are affected equally. Horizontal pedigree pattern, like two will be carrier, one will be affected, one will be fine, right? So that is horizontal. Both males and females, they can transmit the trait to their male female offspring. If both parents are carriers, 1 in 4 offsprings they will have the disease. 1 in 4 will be normal homozygous and 1 in 2 will be normal carriers. Why they will not be affected? Because they need two copies of the same gene to display the trait. The offspring of an affected individual must either have the disease or be carriers, as there is no normal allele. Variable penetrance and variable expressivity are features of autosomal dominant inheritance. So there is an increased risk of autosomal recessive conditions in the offspring of consanguineous marriages. Examples include, this is the most common example, cystic fibrosis, then sickle cell disease, phenylketonuria, androgenital, this syndrome, and mucopolysaccharidosis, and spinal muscular dystrophy. X-linked recessive inheritance. Males are affected predominantly. Male-to-male transmission never occurs. Unaffected males, they never transmit the condition. Affected males, they transmit the allele to their daughters, who become heterozygous carriers, and they are almost always unaffected. Lionization, which means random inactivation of one X chromosome, it may result in some females having mild symptoms. This here like 10% cases of this hemophilia, they can be due to lionization. It means there was random inactivation of the maternally inherited X chromosome. So now the paternally inherited X chromosome is going to exhibit features of hemophilia. So they say night move pedigree pattern, but you don't have to remember it because it will go like night can move in all directions. Okay. So the severity of the disease is uniform across generations. A woman with an affected son and an affected brother is an obligate carrier, and means that if she had an affected brother, or now she has an affected son, so it is confirmed that she is a carrier, and the possibility of two independent new mutations is extremely small. A woman with one affected son is not an obligate carrier. So with one son, we will not say that she is a carrier. Examples include red-green color blindness, Duchenne or Becker muscular dystrophy, hemophilia A and B, and X-linked A-gamma globulinemia. Remember, like B and D, these are X-linked recessive. Due to stress of exam, many students they forget that what is the mode of inheritance. They will remember that there is muscular dystrophy, so this is autosomal dominant. You should remember only myotonic dystrophy is autosomal dominant. Duchenne and Becker, they are X-linked recessive. Okay, and Fragile X syndrome is X-linked dominant. So females are affected by X-linked recessive conditions due to atypical lionization, which means random inactivation of predominantly the normal X chromosome. Then X autosome translocation, under which circumstances the normal X chromosome is preferentially inactivated. Otherwise, an autosomal monosomy would result. For example, Turner syndrome. In Turner syndrome also there is random inactivation of one X chromosome. A new mutation on the second X chromosome will take place in this case. Okay. Another type of inheritance can be X-linked dominant. So males and females are affected. The disease is uniformly severe in males, but the degree of severity is variable in females because of lionization. Male-to-male transmission doesn't occur. All daughters of an affected male, they will be affected. In two sons of an affected female will be affected. Main example is vitamin D resistant rickets. Then what is lionization? We will talk about it in a bit detail because in part 1 they will test you on this one. Random inactivation of one of the X chromosomes in the females and occurs in 15, it will occur usually 15 to 16 days after conception. In interphase cells, the inactive X chromosome appears as a darkly staining mass of chromatin, the sex chromatin or Barr body. Inactivation is dependent on the activity of the exist X inactivation specific transcript, which is expressed only from the inactive chromosomes and results in differential methylation of this chromosome. The X chromosome is, however, not totally inactivated. Otherwise, all females they will have Turner syndrome, and 47XXY or 47 triple X individuals they will be normal. In female carriers of an X autosome balance translocation, the normal X chromosome is preferentially inactivated, otherwise an autosomal monosomy would result and this will be incompatible with live birth. Normal males, they have no Barr bodies. Like 47XXY males, they have one Barr body, and 47XXX females, they have two Barr bodies. Because two X are inactivated, and here one X is inactivated. Okay, and don't confuse this one with Klinefelter syndrome, that is different. Okay. Random inactivation of predominantly the normal X chromosome, it can result in female being affected by an X-linked recessive disorder. Then we were talking about the modes that how DNA can be abnormal. So if you like, you can take a 5-minute break here and then come back. 5 minutes, 2 minutes, just drink some tea, coffee, and then come back. Yeah, yeah, yeah. I will close. Okay, that's fine, no problem. Okay. So non-disjunction. It means failure of the chromosomal bivalents to segregate to the two daughter cells during anaphase. And it can occur during meiosis 1 and 2 or during mitosis. So non-disjunction can occur both during meiosis and mitosis. In which stage? Important to remember, anaphase. Occurring during meiosis 1, the gamete receives both homologues of the chromosome pair. If it occurs during meiosis 2, the gamete receives two copies of one of the homologues of the chromosome pair. The majority of autosomal trisomies they result from non-disjunction during one of the maternal meiotic divisions. The other daughter gamete inherits an autosomal monosomy. This is always incompatible with survival to term. Increased risk of non-disjunction with increasing maternal age. You should remember the risk will increase. And why you will not see many patients with these conditions? Because such fetuses they will be aborted during early phase of development. That was non-disjunction. Now you must have seen in the exam reciprocal translocations. Okay, this concept is not so hard. Just remember that they result from breakage of at least two metacentric chromosomes. Metacentric, where the centromere is in the middle, with exchange of genetic material. So long arm of chromosomes 11 and 22 more commonly involved, like in cases of Lee-George syndrome. Incidence in general population is 1 in 500. The total number of chromosomes is maintained at 46. Individual with balanced translocation, phenotypically they will be normal. Segregation at meiosis may result in offspring inheriting an unbalanced chromosomal complement, resulting in an increased risk of miscarriage or congenital anomalies. Risk of having abnormal offsprings dependent on the particular chromosomal rearrangement but lies in the range of 1 to 10%. These are reciprocal translocations. Another form of translocation is Robertsonian translocation. So here, if you have noticed, the change took place in metacentric chromosomes, at least two metacentric chromosomes. Here, Robertsonian translocation, it results from breakage of two acrocentric chromosomes at 13, 14, 15, 21, 22 at or close to the centromere with fusion of long arms and usually a loss of the short arms. So acrocentric chromosome. Look at this one. This is the parent's chromosome. Okay. Here the chromosomes in parent carrying a balanced translocation, Robertsonian translocation. Some one, ideally they should have been like this is in the makeup in one. Okay. This is in the other one. Okay. Chromosomes in the other parent. This is the normal arrangement. But what happened that this bound region, instead of lying here, it has gone over the top of another one. Okay. But still the amount of DNA is the same in the system. Then if you will get two parents like this combination, what arrangements you can see in their children? They can be absolutely normal, like this one of the parents, or they can have a balanced translocation, like the same amount is there, but it is on top of another one, or then it can be unbalanced. Why unbalanced? Because it is not a variation of this one or this one, okay? This one is additional, or this brownish, the short arm, additional short arm. So this type of abnormalities will be there. This one will be balanced because it is just like the parents, only arrangement is little bit different. Here you have got additional one and here additional short one. So the total number of chromosome reduced to 45. Yeah, so short arms usually contain only ribosomal RNA genes with multiple copies on other acrocentric chromosomes. Multiple copies, like this is an additional copy here. Incidence is usually 1 in 1000. 13q and the 14q translocation most common between the long arm of 13 and 14, and that will be exhibited in Down's syndrome. Increased risk of miscarriage, fetal anomaly in carriers of balanced Robertsonian translocations. In the exam, they will try to confuse you by giving you unbalanced. You should remember it is in the balanced Robertsonian translocations. Female carriers of 13q, 21q, or 14q, 21q Robertsonian translocations, they have a 10% risk of having a baby with Down syndrome, while male carriers, they have a 1 to 3% risk. So 10% if the female is affected and 1 to 3% if the male is affected. And this is also SPA question. If there is balanced translocation in 21q, then the carriers, they have a 100% risk of a live birth with Down syndrome. So monosomy 21, which is the other possibility, is not compatible with live birth. So here the chance will be, if they give you this combination 21q, the chance of recurrence of Down syndrome will be 100%. And if they give you this translocation, then you should remember if the female is the carrier, mother is the carrier, then 10% chance of having a baby with Down syndrome. The recurrence risk will also be the same. And if this combination is seen in the father, then the risk of recurrence will be 1 to 3%. And with this 21q, 100% risk of a live birth with Down syndrome. Then chromosomes, they can show mosaicism. It means the presence of two or more cell lines within an organ. Individuals with different genetic constitutions but derived from the same zygote. So the zygote will be the same, individuals will be different. So the presence of different cell lines derived from different zygotes is termed chimerism. If they are from the same, then it will be, if they are coming from the same, that is mosaicism. If they are coming from two cell lines, that will be chimerism, and may occur, for instance, as a result of twin-to-twin transfusion syndrome. Mosaicism usually results from mitotic non-disjunction in an early stage embryo, and mosaicism is seen in 1 to 2% of all cases of Down syndrome mosaics. So that's birth, and it forms the basic concepts of this genetics. Now we have to talk about other abnormalities. So previously, when we used to talk about fetal anomalies, it was found that the risk of background risk, I mean the risk in the general population of getting some abnormalities is 1 to 3%. But later on, the studies have shown that birth defects or fetal anomalies are common, with 2 to 3% of neonates having a major abnormality and up to 10% having minor abnormalities. A useful way of considering anomalies and how they may have occurred is demonstrated in the following classification. Malformation. It means it has resulted from inherited abnormality in early embryonic life. For example, congenital heart disease. Disruption type of fetal anomalies as a result of external pressure on the fetus. For example, talipes in oligohydramnias, since there was very little liquor, so the baby developed talipes, which is the bending of the foot, because there was no liquor, couldn't move, so there were amniotic bands and the normal pattern was disrupted. Then deformation. It means abnormal developments as a result of external interference. For example, teratogens, maternal infections, or trauma, for example, from amniotic bands. Amniotic bands, like in cases of Porter's syndrome, when there was renal agenesis and there was no liquor. So what happened? Amniotic bands were formed around the baby. So baby developed a certain postural abnormality. Then dysplasia. This results from abnormal tissue development. This can either involve a single organ, for example, schizencephaly, involving the brain, or an organ system, for example, osteogenesis imperfecta. So many of the dysplasias, they are genetic in origin. For example, mutation of the CoA1 gene in osteogenesis imperfecta and the association of EMX2 mutation in some cases of schizencephaly. So osteogenesis imperfecta, the baby will have brittle bones, bruised sclera. So then what is a syndrome? A syndrome refers to a collection of abnormalities whose pathogenesis is linked. For example, Down syndrome resulting from a chromosomal abnormality. Then Meckel-Grubert syndrome resulting from a single gene disorder or collection of abnormalities relating from environmental exposure to teratogens. For example, in cases of fetal alcohol syndrome, there is just one teratogen, but it has caused many abnormalities in the baby. Then sequence defects. A single defect can have a cascading effect on other defects. For example, in cases of Porter syndrome, like there was oligohydramnias because the kidneys they were not producing enough urine. So as a result, there was talipes, hip dislocation, and lung hypoplasia because there is no life there. Nothing is there, so baby is going to have these problems. But the later it has been shown that if there is oligohydramnia, it is not going to cause lung hypoplasia. Okay, but it can occur in cases of Porter's syndrome. So whenever an abnormality is detected as part of antenatal ultrasound screening in the mid-trimester, consideration should be made to the possible underlying genetic causes for the abnormality or abnormalities found. This may involve offsprings like couple karyotype assessment of the fetus by amniocentesis. This may involve offering the couple karyotype assessment of the fetus by amniocentesis or CBS to diagnose chromosomal abnormalities. Alternatively, samples may need to be sent for DNA analysis in the event of a possible single gene abnormality. The couple may benefit from referral to the clinical genetics team for further advice and counseling. So, then some of the anomalies they will have a multifactorial inheritance. Many common diseases and malformations they show a familiar tendency, but they do not follow a simple Mendelian patterns of inheritance. These conditions are said to be multifactorial in origin. They are more common than single gene or chromosomal abnormalities, and they include cleft lip and palate, talipes, congenital heart disease, and neural tube defects. The X-linked inheritance, autosomal dominant, recessive, and other two types, they will follow Mendelian pattern of inheritance. So multifactorial is not following the Mendelian pattern. Okay. So the multifactorial conditions are also seen in adulthood, for example, diabetes and cancer. The conditions are considered to be related to the effects of gene abnormalities in association with environmental effects. So how do we do clinical assessment of mode of inheritance? In a couple presenting with or positive family history of a condition, a family tree should always be taken. This may enable the pattern of inheritance to be identified and therefore allow individual risks to be quantified. So even for the sake of exam, you have to draw the pattern, okay? Don't try to answer them blindly because you need to answer them correctly. Okay? If you remember, like they are asking you about the simple reproduction that this syndrome is going to cause what, then you can do that. Otherwise, if you are in confusion, just make the pattern. Okay. Sometimes, but usually in the exam they will give you a picture in which they will show you that this is the question. So what is the mode of inheritance? Okay. They will draw the pedigree themselves and they will just show you the affected individuals and they will ask you about the modes of inheritance. So, so just for the sake of remembrance, just remember autosomal dominant conditions they include achondroplasia, adult polycystic kidney disease, adult, okay, Ehlers-Danlos syndrome, familial adenomatous polyposis coli, familial hypercholesterolemia, Gilbert syndrome, Huntington's chorea, von Hippel-Lindau disease, Marfan syndrome, myotonic dystrophy, neurofibromatosis type 1 and 2, tuberous sclerosis, von Willebrand's disease, breast ovarian cancer susceptibility, like they will carry BRCA1 and BRCA2 genes. Autosomal recessive conditions, I want you to learn them by heart because these are very important because definitely they will ask question about the modes of inheritance or what is the mode of inheritance in this condition. Autosomal recessive conditions they include, as we know that they will cause metabolic disorders, alkaptonuria, alpha-1 antitrypsin deficiency, ataxia telangiectasia, beta thalassemia, congenital adrenal hyperplasia, cystic fibrosis, Steuben-Johnson syndrome, Fanconi's anemia, galactosemia, glycogen storage disease, homocystinuria, hemochromatosis, oculocutaneous albinism, phenylketonuria, sickle cell anemia, spinal muscular atrophy, Tay-Sachs disease, and Wilson's disease. Then there are certain X-linked recessive conditions like Becker's muscular dystrophy, Duchenne muscular dystrophy, Fabry disease, Fragile X syndrome, hemophilia A and B, Hunter's syndrome, ocular albinism, red-green color blindness, testicular feminization syndrome, and Scott-Eldrick syndrome. Then X-linked dominant conditions, these are and because this is uncommon mode of inheritance, like vitamin D resistant rickets, incontinent pigment, and then Rett syndrome. Then there are certain Y-linked syndromes also. So before we didn't know about many X-linked, Y-linked syndromes, but now we know that there are many Y-linked syndromes also. So there is little genetic information on the Y chromosomes other than that for gender determination. One gene that codes for hairy ears, it has been identified on the Y chromosome. So that is the only condition we know about the Y-linked chromosome. But there was an article in the TOG and also in the current Journal of Obstetric and Gynecology. They said that all conditions where before they didn't have any clue to their mode of inheritance, they said that like Prader-Willi syndrome, Angelman syndrome, Beckwith-Whiteman syndrome, all of these have been attributed to changes in the Y chromosome. Similarly, Young's syndrome, it is also due to some problem in the Y syndrome. They will be relatively infertile. Okay, that is the point. So, an autosomal dominant condition, the trait is demonstrated in individuals with one normal allele and one mutated allele. So, the offsprings of an individual with an autosomal dominant, they have a 1 in 2% two chance of like 50% chance of being affected. Now we are revising in the concluding in the end. Then autosomal recessive conditions are only manifest as the full phenotype when the individual is homozygous for the mutant allele. Individuals who are heterozygous for the condition, they often show no features and they are completely healthy. In X-linked recessive conditions, the mutant gene is carried on the X chromosome and the condition is usually only manifest in males. Healthy heterozygous females, they transmit the condition to one in two of their sons and one in two of their daughters. They will become carriers. X-linked dominant conditions are uncommon and the conditions are manifest in both males and heterozygous zygote females. That is the that's all about the patterns of inheritance and the basic. It forms basics of genetics. So please take a 5 minute break here. Then we will talk about specific syndromes. Just take like 4 minutes, or 5 minutes, and then please come back. Okay.